This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 304, recorded on September 26th, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today here in studio, Dickson Despommier.
Hello, Vincent.
How have you been, Dickson?
I've been great.
Really great?
Yeah, thanks for asking.
Not that I care, you know. What's the weather today, Dickson?
It's beautiful. It's about in the 70s, low 70s. The humidity can't be above 40%, and there's no wind to speak of. And it's very pleasant outside. We should have had this podcast outside.
It is sunny, no clouds.
No clouds.
23 Celsius.
Yep.
48% humidity.
Oh, that was close.
You got it wrong, Dickson.
Close.
You shouldn't be talking about the weather if you don't know what you're talking about.
Is that what our show is going to be about today?
How did you know, Dickson?
I kind of guessed by this morning's banter.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
How's it out there? Raining?
No, it's beautiful, sunny, everything's great.
Right.
Wonderful. And from... From Western Massachusetts, Alan Dove.
Good to be here. I guess it's kind of south-central Western Massachusetts.
No, don't do that to me. I have enough geographical issues.
It's just not Eastern Massachusetts.
No.
Western Massachusetts.
Western.
Yeah, West is good. I mean, that's what you told us from the start, so let's stick with that. We have a guest today in studio. He is a professor here in the School of Public Health. It's called the Mailman School of Public Health. It's actually a separate school up here at the Medical Center. He's in the Department of Environmental Health Sciences. Jeff Shaman, welcome to TWiV.
Thanks, Vince. Thanks for having me here.
My pleasure.
If I can add, I'm about 3 feet north of Dickson right now.
And what's the weather like over there, Jeff?
Yes, he is. He's right next to Dickson. He's not in his own office. Easier to do it this way, so
Right.
we can see you. Well, you're just right across the street from us, right? Anyway.
Yep.
So we brought Jeff over because he is a epidemiologist, and he's going to help us talk about Ebola. But before we do that, we have a couple of things on the list first. As soon as my Google Doc unfreezes. Well, this episode of TWiV is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. And this is a wonderful institution here in New York City. Over 20 virology labs, all in one building in New York City. The department's a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit graduate students to apply to their program by December 1st. That's the deadline. So if you want to do virology, microbiology in New York City, apply to the program. Postdocs, too. If you're interested in joining this department, check out the faculty list and email them directly. For more information about the department, please visit mssm.edu/MIC. You know, whenever you do an ad for something, it's always nice when you can say you actually use the product and can endorse it. So I used this product. I went to graduate school at Mount Sinai. I got my PhD there with Peter Palese. So I endorse it. I vouch for the excellence of that program. Another item of business today, at the American Society for Microbiology, they have a communications fellow position opening up. If you're an early career scientist interested in public outreach, you love microbiology, this is a fellowship at ASM's headquarters in Washington, D.C. It's a good opportunity to learn more about science communication. They'll teach you how to do it. It's a 6-month fellowship, and they are now accepting applications through October 22nd. There is an online application form at bit.ly/communication fellow, all one word, bit.ly/communication fellow. Now, Jeff, you've been here a couple of years, is that right?
3.5 years.
Where were you before?
I was at Oregon State University. I was a professor of atmospheric sciences there.
Oregon State in
Corvallis.
So
Wow.
that's the eastern part of the state, right?
No, it's in the Willamette Valley. It's about an hour and a half due south of
I'm
Portland.
thinking of Eugene. That's in the east.
No, that's actually another 45 minutes due south. They're all in the Willamette Valley.
I shouldn't talk about geography. I obviously don't know anything about it. Yeah. So you were a professor there, and before that, what were you?
Well, before that, I did a postdoc at Harvard at the Earth and Planetary Sciences and at the School of Public Health there. And I did my doctorate here at Columbia in the Department of Earth and Environmental Sciences. So I'm actually a geophysicist by training. My PhD is in climate and atmospheric science and hydrology. But I've had a lot of interest in infectious diseases and trying to model them and to look at the environmental determinants of them, particularly hydrology and meteorology and how the variability of that affects the transmission dynamics of those systems.
You know, I knew it was only a matter of time before we got a meteorologist on the show.
Well,
Sorry.
maybe he can explain relative humidity to us.
So you know all about the weather then?
I know a lot about the weather, that I do. And
But you can't do anything about it, is that it?
Yeah, well, you know, that's one of the interesting things about it. You can predict the weather, but you don't change it. Maybe you can predict infectious diseases, but you can actually change the outcomes.
Right.
It's true.
That's
So-
true. That is true.
So now you focus on infectious diseases, predictions of outcome. Is that?
Yeah, well, you know, I've even in my doctorate work, I was doing a lot of work on infectious diseases, mosquito-borne diseases, and I was looking a lot at the environmental determinants of disease. And I was interested in and eventually branched into looking at respiratory diseases, influenza. And that led to working a lot on modeling them. So a lot of my work in climate and atmospheric science involves a lot of computational methods, a lot of statistical methods, and mathematical methods combined. And I've just begun applying some of those methods and taking on some of the methods that are typically applied also in epidemiology and use that to actually model these systems.
Great. Well, I'm hoping you can help us with Ebola modeling today. And before we get to that, we do have some follow-up as usual. And as usual, we've done Ebola for the last couple of episodes. And as I said last time, it generates more follow-up than almost any other topic except maybe XMRV. First email is from Rich Condit, I presume, right, Kathy?
Yes, I think so.
R.E. John's E.E. Cummings letter. John writes of fear of science and implies mistrust of science and scientists on the part of some lay people. He points out that critical thinking does not necessarily require a science education. He writes of our ivory tower. Conflating these and other comments, I am reminded that at various times on TWiV, we have made offhand comments to the effect that so-and-so is not qualified to evaluate or understand this or that because he is not a scientist. This sort of statement sets scientists apart and isolated from non-scientists, which is exactly the opposite of what we are trying to accomplish with TWiV. We want to be inclusive and have everyone join in the fun and insight that science brings. John is dead right that one does not have to have a specific training to be a critical thinker or even to understand in significant detail complex scientific concepts. We see this all the time in the correspondence we receive. So thanks, John, for the reminder that we are all just humans and that our mission should be to help each other through this life with whatever gifts we have been given. I think that's a good sentiment, Rich. Thank you. And he's right. We try not... anyone can have an opinion. Absolutely. And we should avoid saying you're not qualified to have
I do
an
have a
opinion.
problem with uninformed opinions, though.
Yes.
It's not that somebody is not capable of understanding something, but when someone puts on a pretense of knowing what they're talking about and clearly doesn't, that is a problem.
Yes, I agree.
But yes, this is not a scientists versus the public type of thing. This is you should study an area and pay attention to what's going on in that area before you go and make statements about it.
I mean, we have sometimes called people out who are scientists and say, maybe we say, you don't know what you're talking about. But
Yeah.
we don't usually say that of the public, I think, right? Because we want to embrace them and participating in this discourse.
Absolutely.
All right. Alan, can you take Robbins?
Robin, this is the discussion about aerosol versus droplet. When a fixed-wing aircraft on its rollout reaches rotation speed, it transitions from wheel-borne to wing-borne. Once it's fully wing-borne, it is airborne. The interaction with air is at the surface of the object. The surface decreases by the square root, while the volume and mass decrease by the cube root as objects get smaller. Beyond a certain point, even without wings, the surface area is adequate for the gentlest of air currents acting on it to produce enough force to keep the object airborne, long beyond the ballistic trajectories produced by forces acting on projectiles. The sol suffix in aerosol refers to particles in suspension, borne by the medium. The gentlest of currents in the medium delay the settling out of the particles. The particles in a spray are initially all ballistic, like a ball that is thrown. If they're small enough or can become small enough soon enough through evaporation of contained liquid, then they can transition from ballistic to airborne, much as an aircraft catapulted off an aircraft carrier transitions from ballistic to wingborne or airborne. In the case of the particle, the air currents exert enough force on its surface area to support its mass. In the case of the aircraft, the thrust of its engines increases its airspeed, and thus the force exerted by the air on its wings. Sorry if this seems like preaching to imbeciles. Some folks, like some bullfrogs, may still not get the difference between a ballistic droplet spray and an airborne aerosol.
We've been having a discussion about aerosols and airborne transmission for the past couple of weeks, so this is one of our listeners weighing in on that. I think we've defined aerosol well by now,
yeah.
Right?
And I think when we say airborne transmission, we mean not droplets. We mean something that if I'm sitting next to you breathing it out, you will breathe it in.
Right.
So that's the distinction that we've drawn.
Kathy, can you take the next one?
Sure. Cameron writes, "Hi guys, great show. I listen to you guys every week and love what you do. I especially like that you counter all the misinformation in the media currently that Ebola could just so easily mutate and become airborne. I'm tired of hearing how, since the resistant strain of Ebola can transfer from pigs to primates, that it demonstrates that Ebola can become airborne, especially when it doesn't then spread between the infected primates. Not to mention the fact that the resistant strain isn't lethal to humans. It is also relevant that the Russians tried to weaponize lethal strains of Ebola into an airborne pathogen and failed. I blame movies like Outbreak for the fear in the general population that viruses just decide to change their mode of transmission, even when it has not been observed in the real world before. Anyway, the questions that I do have about this particular epidemic are, it is my understanding that Ebola as a zoonotic virus may infect other mammals, such as dogs, without them necessarily becoming symptomatic of the infection. Since the current situation in countries like Liberia, where carcasses of the deceased are being left lying around and assuming that they may be eaten by dogs, birds, and other carrion-feeding animals, what would be your view as to the possibility of developing new reservoirs of the virus, in a similar fashion to the crows becoming a reservoir to the West Nile virus in the U.S.? Is it your opinion that in the long term this outbreak could be more difficult to control than the current estimates would assume? Or do you think that the possibility of new reservoirs of infection is insignificant? It would seem to me that this current outbreak is a little different from past outbreaks, since the number of cases have overwhelmed the medical response and have affected the ability of authorities to even collect the dead from some regions. By the way, I agree that mosquitoes are virtually zero risk in spreading Ebola. Cheers from down under. Cameron. The comment about Ebola virus in dogs, there's an article that I found cited in Wikipedia that describes the fact that dogs can become infected with Ebola virus. There's a study in Gabon, and these researchers published in the CDC-based journal Emerging Infectious Diseases, where they found that dogs do eat infected animals and corpses of humans and remain asymptomatic, but they can have a high seroprevalence for Ebola virus. And it's sort of distance related. The farther away they are from an outbreak, the lower their seroprevalence with antibodies for Ebola virus. So the strange thing that I found in that paper, I didn't read the whole paper, but I looked at the abstract. The strange thing to me was that they cited 2% Ebola virus IgG in samples from France.
Yes.
And so that made me a little wary of their assay, but
I think that's probably going to be the background of their assay.
Yeah.
because it's
There is a former French colony in West Africa, however. So the Cameroons and Côte d'Ivoire. So it's possible that during some time they collected sera from people who were asymptomatic.
These are dogs, Dickson.
Well,
These are dogs.
And from dogs that were asymptomatic from those regions that were brought back to, France.
no. I think
pets.
these are samples that they took from dogs in France.
Yeah.
I believe so. Yes.
Okay,
Yeah.
okay.
So that's a little more problematic.
Okay, that's very problematic.
Well, I don't know what the likelihood is that it would establish itself in a dog or any other animal. I mean, who knows, right? The fact that you have antibodies in dogs suggests that the virus is replicating.
Is it cross-reacting with some other agent, perhaps?
It might. So Jens Bossi, who we had on a while ago, or Kuhn, I forgot his name.
Jens Kuhn.
Oh my gosh. He would say it's probably cross-reactivity, but I think you need to do more of these sorts of studies, right, to figure it out.
Right. I mean, the result from France suggests cross-reactivity, but then why do the percentages go up as you get closer to Ebola?
Yeah, that's actually a good control, right?
Yeah.
I think you need to keep looking, and especially in the current outbreak, right? But right now, people aren't concerned with serosurveys, right?
Right. Yes.
No.
Well, that's not the immediate concern, but certainly understanding zoonotic transmission of this disease, the reservoirs and the spillover is important. They know it's in bats, they know it's in rats, they know it's in primates, dogs, pigs. These are things people are concerned about because of carrion eating.
Absolutely.
So, Jeff, you think after this subsides that people will do some serosurveys and get information on that, and also if people are infected asymptomatically, for example?
Yeah, I think that needs to be done, and I think it certainly will be done. I mean, there have been some surveys done in Central Africa looking at, in particular, there was a paper out looking at why has there not been a documented outbreak of Ebola in the Central African Republic, which is sort of ground zero for the whole nexus of where activity typically took place prior to this West African outbreak. And they found positive rates of about 10% of the population. In the pygmy population, it was 13% had antibodies, and in the non-pygmy population, it was about 10%. And so then that begs the question, what's going on? Is it cross-reactivity? Is there some lower pathogenic strain out there that's doing this? What is it?
Or a unique competent region of their MHC allows them to resist more than others.
All right, the next one is from Dawn, who writes, "Greetings, I'm very happy to have learned of your podcast. However, I was really concerned while listening to your recent podcast on Ebola. Specifically, I was incredulous that your 3 hosts did not even know the incubation period of Ebola. I'm glad you looked it up. I was dismayed how you seemed to disregard downgrade transmission via semen. Incubation to infection to transmission can vary from a few days to several weeks. One of your hosts seemed to suggest that any man exposed to the virus would immediately not be inclined towards having sex at any time before he gets sick or during the long convalescence, even though he's potentially infectious for up to 2 months. Come on." I was dismayed by this discussion. Sex and prostitution are rampant all over the world and prevalent in Africa. As a public health nurse, I could almost not believe what I was hearing. You almost seem to disregard sexual transmission of Ebola, which is near criminal in my opinion. I was also kind of nonplussed by your flippant, chatty treatment of this disease that is killing hundreds. Although I can understand it, health professionals have a whole new level of black humor. I realize you're doing important research. I do love your podcast. I know you're not interested in epidemiology or the broader social issues. So I won't talk about the weather here in North Carolina, nor will I ask you to be more PC or more careful where Ebola is concerned. Except, damn, for the host to have to look up vital info on the CDC site, simple facts that, capital letters here, you don't even know. Maybe stick with things you understand more, i.e. not Ebola. Or pick other topics, respectfully. So this is quite offensive to me. So let me just try and be calm and address some of the points. So someone asked, I think Dickson asked, what's the incubation period? I said 14 to 21 days. And I said, but let me look it up. And it was 2 to 21 days. So I don't know why we need to know the incubation periods of all disease. Like a scientist, we knew where to look it up.
Right.
And I have a real problem with chewing people out for checking their facts.
Yes, as a science writer, absolutely.
Right, so, yeah.
Now, the discussion about sex. So we talked about how the virus is found in semen for a while after infection, after the symptoms subside, and we speculated that you might not be interested in having sex because I understand that you don't feel well for quite a long period after Ebola infection. CDC does recommend you wear a condom after having Ebola if you choose to engage in sex.
Right.
So that's perhaps the way we should have left it.
Yep.
We are not flippant or chatty about the disease. I think we're quite serious, and we've devoted several episodes to it because it's a serious business.
And I'm pretty sure we've touched on some of the broader social issues quite a bit.
To say we're not interested in epidemiology simply isn't fair, because when even basic virologists such as Kathy and I and Rich Condit are, when we talk about disease, we have to incorporate epidemiology in that discussion. Now, I know we are not trained as epidemiologists, but to say that we're not interested in it, I think, is simply unfair.
We've had a lot of discussion about the ecology of these things also, which fits in very well to the natural history of these diseases, and I think it's really should probably go back and listen to all of our TWiVs.
Dickson, do you remember the incubation period for all of your parasitic diseases?
Most of them.
Yeah?
Most of them.
And that's because you taught it for years?
Yeah, since 1971 until 2009, so you don't tend to forget things like that. But you go over them every year, by the way. You don't take it for granted that you know it. You have to review the literature every single year, and you have to bring your facts up to date. So if it changes, or if a new strain comes in that has a different number, you're obliged to learn that. So the answer is yes.
So one more statement here, and then we'll move on. You almost seem to disregard sexual transmission of Ebola. As far as I know, there's no evidence for sexual transmission.
That was our statement. That was our statement.
Is that? Would you know, Jeff, if there's any evidence for sexual transmission?
There is one piece of evidence that I read, and I wouldn't be able to tell you the source right now, where it stated that the virus was detected in semen of a man 60 days post-recovery.
Right. But whether he transmitted it.
But whether he transmitted it and whether they've actually documented that, I don't know.
Sure.
Or whether he could transmit it. Because I think we talked about the same paper came up, and wasn't the 60-day sample was PCR positive, or was that we discussed some issues about PCR positive versus culture positive as well.
Yeah.
Right. I'm looking up the link for that paper again. I believe this Journal of Infectious Disease paper. Right. Bausch was the first author, John Towner, Scott Dowell, Felix Caruso, Matthew Lucuia, and, sorry, I'm slaughtering the names, Anthony Sanchez, Stuart Nickel, Tom Kizek, and Pierre Roland. And that paper was linked to in last week's episode. And you guys kept quizzing me and making me look further and further into the data. And, but yes, the semen samples were in table 3. Sorry. I'm fact checking. Sorry. Takes
yes,
a little
exactly.
time.
Sorry, Dawn. We're checking our facts.
Exactly.
Yeah. Rats. I'm not finding the right table. But I'm pretty sure what we found was that the semen was virus positive, not just PCR positive.
PCR positive, even at the 2-month mark.
Right.
So
the issue then is, if this is a sexually transmitted disease, why aren't we seeing sexual transmission of it? And you can come up with a whole bunch of explanations, but there it is. We don't have clear evidence for sexual transmission.
Of course, we shouldn't dismiss it. I think we were probably saying it couldn't happen because people aren't going to be feeling up to it. But I understand that, especially in an outbreak of this size, you don't know what's going to happen.
Sure.
So you have to be careful.
One other thing I wanted to mention is that we have had epidemiologists on the show even before today. We had Tyler Sharp.
Yes.
Or it was before my time even, but it was TWiV 146, I think.
Right.
So we definitely have an interest in epidemiology.
And we had Mike from Brooklyn.
That's right.
Do you know Mike from Brooklyn? What's his last name?
Oh, yes.
Mike, the epidemiologist from Brooklyn.
Yeah, he did a very good job of explaining.
He was great, and we should also have him back from time to time. I can't remember. Right. I'm sorry, Mike.
Infection Landscapes is his
Yes, that's
site.
Right.
I remembered his site name and I forgot his last name.
And I did find this table again. It was table 1. The semen was virus culture positive in 50% of samples tested, which was 2 evidently.
Wow.
In one line of data, and I'm not going to go through all the rest of it.
Okay.
Mike Walsh.
Mike Walsh, right.
Bingo.
Sorry, Mike. We forgot you. We remembered
So,
you.
you know what we're going to do? We're going to edit all of this, and it will appear as though we remembered it.
So that's the other thing. I don't edit, so you hear us making mistakes
Right.
and looking up data, because I think that's important for people to know,
Exceptional.
right?
It's
And-
kind of like being transparent about how one does science.
Yeah.
You can't pretend that you know everything, because you don't.
And my thought, even when Vincent was looking it up on the CDC website, was that he was sort of teaching the audience how to fish, if you will go with that analogy.
Yeah.
Instead of just giving them the fish, he was teaching them, you can go to the CDC website and look these things up.
Here.
Now, and I just want to remind everyone that we were chided for not talking about Ebola a month ago.
Yes.
And that's why we're focusing on it, and we recognize that it's important and that you depend on us for information. Anyway, let's read one more about Ebola. Alan, can you take Thelos, please?
Okay. Thilo says, "Hi, Vincent. First of all, thank you so much for your podcast. They keep me sane and educated in the traffic jams on my way to work. I have a strange question, but do you happen to know how it might be possible to get involved in the efforts in the Ebola outbreak? Currently, I'm unfortunately not in any institution that is even remotely involved in aid work. As I'm UK-based, asking you from the other side of the lake maybe does not suggest itself, but so far I had no response from the charities, Red Cross, MSF, etc. Anyway, a bit about myself. Currently, I'm a PhD student, biophysics for some reason, but have previously gotten a BSc with honors in infectious diseases from the University of Edinburgh. Further, I've been on a 1-year course in forensic science and medicine. Before joining university, I was working as an ambulance technician for the German Red Cross. Currently, during the summer months, I'm a beach lifeguard for the Royal National Lifesaving Institute in Scotland. Already during my time at school, I've been in Lambaréné, Gabon, as a volunteer for the University of Tübingen in Germany to collect blood samples for a research project about malaria. It would be great if you could let me know if you had any idea how I could go about that, or if you know anyone who might have any use for myself. Thanks again for your podcasts, and keep up the work, as I'm always looking forward to them. Thanks a lot. Thilo, P.S. As you might suspect, here in Scotland, it is windy, wet, and cold. I actually have a little bit of insight on this. I've done some disaster services volunteering with various organizations, the Red Cross, the U.S. Coast Guard Auxiliary, and my suggestion would actually be, rather than, it's true when you see some disaster unfolding someplace and it's in the news and you see it all the time, you want to go and help those people. And I totally understand and applaud the motivation. But that's not the way the disaster response works, at least not when it's working properly. So the thing to do would be to approach an organization like the Red Cross or Doctors Without Borders, and instead of saying, can you send me to Africa to help with Ebola, say, look, I'm interested in volunteering for your organization in disaster services. And if you do this with the Red Cross, they probably with Doctors Without Borders as well, they will have specific requirements and training that they will expect you to fulfill as a volunteer. This is not just a silly requirement. This is absolutely essential, because if you don't understand how the organization works, you are worse than useless in a disaster zone.
Right.
So the very first thing to do, and with your experience in the RNLI, you probably understand this. You know, if somebody shows up to the RNLI and there's somebody out in the water drowning, they say, hey, can I swim out and get them? And no, that's not how the process works.
Then I'll have to rescue 2 people.
You cause more casualties that way.
Yeah.
So the way to do this is to go through the front door of these organizations, and you should expect them to probably not assign you to the specific disaster that got your attention in the first place.
Yeah.
With the Red Cross, you will most likely end up staffing a temporary shelter that's been set up for people who were burned out of their building in Edinburgh. All right, doesn't sound very glamorous, but it's essential work. These are people who've been burned out of their building in Edinburgh. So that's the kind of approach to take. But by doing something like that, you've maybe freed up somebody who is I don't know, a registered nurse who's fluent in several African languages, who they can now send to Sierra Leone to work on the Ebola outbreak. So
Yep.
that would be the approach to take, I think.
Another option is to get in touch with the London School of Hygiene and Tropical Medicine and find out who's involved in this over in Africa and associate with those people, because you've got science training already and you can speak the language. So they should be able to find, a niche for you somewhere if they're already engaged. So that's another possibility.
I'm going to get back to the follow-up later because I promised Jeff that he could leave at 3 o'clock.
Okay.
He's not feeling well anyway, so I want to get his take. The reason I asked him to join us, the CDC this week released, as part of Morbidity and Mortality Weekly Report, a document called Estimating Future Number of Cases in the Ebola Epidemic. And in the references was a link to Jeff's website called Prediction of Infectious Diseases, and you have Ebola and influenza there, and you have a graph which you can change the parameters to all the countries: Guinea, Liberia, Sierra Leone, and all infections, mortality. So I wonder if you could, the numbers that CDC came up with were scary, and the press was going crazy because their long-term prediction for the end of January, I think, is between 500,000 and 1 million total cases. And that ranges because there's some underreporting going on. So I wonder, Jeff, if you could tell us how you make these kinds of predictions. What kinds of data do you use, and what kinds of math, basically?
Well, there are a number of ways of approaching it. The outbreak is scary right now because it's been reported in the media. It's growing exponentially, and that's a multiplicative effect as it gets larger each week. You go from 1 to 2 to 4 to 8 to 16 cases, something along those lines. Not quite at that rate, but it means that there's a rate of doubling that goes on. And if it takes 30 days to double, then you double again after another 30 days, and that's an exponential growth period. Because of that, and because it's had that behavior, you could almost do something very trivial, which is just plot the incidence on a log linear plot and draw a line through it and make a projection. Now, the issue with it is at some point it's got to turn around, one would think, all right? Certainly within a country like Liberia, ultimately, there's a cap because you're going to run out of people to infect. Right now, we haven't run into that. You haven't had any issues of herd immunity, as we think, because there's almost no seroprevalence in the community there, and we think that the community at large is largely susceptible. But embedded within it, the disease is very heterogeneous. It's very patchy, and there could be local clusters of outbreaks where you can run into some herd immunity, and that may change things. So very, very simply, just looking at it, you see sort of this unlimited untapped potential within the country where you have tons of people that you can infect over time, and you see this very simple growth pattern that's an exponentiation. So you could use very simple methods to it. We tend to apply more sophisticated methods also to it. So some of the methods that we do, we use a combination of what's called dynamic modeling, where we have a model that represents the propagation of Ebola through a population. And we use the observations of what's happened in preceding weeks up to the present to optimize that model. And there are all sorts of strategies that are used to optimize it, but ours are based actually on numerical weather prediction. It's the same way that they optimize a numerical weather forecast model prior to actually generating the forecast. And we do the same thing, and then we generate these outcomes. And what happens through this process of optimization or data assimilation is that the state of the model, how many people are infected now before you make your forecast into the future, is better constrained, you hope. And all the things under the hood that we would like to have a really good understanding of are hopefully estimated. Things like the serial interval, the basic reproductive number, which is a measure of the force of transmission. And if we have those better constrained, we're going to have a better estimate of how the trajectory will unfold in the future. And that's what we then feed into that prediction. That said, the Ebola environment is very challenging for a number of reasons. Firstly, we've certainly never had an outbreak of Ebola of this magnitude, and we've never seen it in densely populated settings. And that may change a few things. That can alter things a little bit. Secondly, the information that we have on the ground is unfortunately very incomplete. So we really only have bulk numbers. We have delays in reporting. There's a suspicion that there's some serious underreporting going on, which biases the data low, which is not terrible for the modeling projections. It just means your model projections will be biased low. More problematic is the fact that the information that comes out about new cases for this week may actually represent cases that occurred weeks before. And that may mean that you really need to pile them up before, and that there was a steeper growth of the outbreak than is really evident in that data. The WHO does have line data. They're not sharing it broadly. There is a collaborating center, and they published in the New England Journal of Medicine this week where they did modeling with that. And part of what they were able to estimate with that are some facts that are a little scarier than what you would look at just looking naively at the incidence and mortality rates. If you just look at those naively, you can see case fatality rates of 50%, maybe even down to 30% in parts. But when they looked at the line records, which show this person, did they recover, did they die, what happened to it, they're finding that the case fatality rate is really 70%, and that there's some significant underreporting and also what's called right bias in the whole thing, that we haven't gotten to those who have been infected, they haven't all died yet. So in that sense, it's very frightening. And in order to inform these models, better data is better. Now, that's not the priority right now. The priority right now is boots on the ground. It's nurses and doctors and beds and resources and security. And that has to be put there. But unfortunately, the rules of how they engaged Ebola previously in small isolated villages are not working here. And that involved a lot of epidemiology, in fact. I mean, if you talk to Doctors Without Borders, they would go in and they'd set up an isolation area and their CRU center. And next thing they do is they go out and they do contact tracing, where they take every patient and they find out who were they in contact with. Well, given the magnitude of this outbreak, that's not feasible. And so you're stuck with not being able to figure out who is potentially at risk, to monitor them, watch their movements, and monitor whether or not they're going to come down to it. Because it's not in a small, isolated setting anymore. It's in the cities. All those things make the control more challenging. So what you're left with are these crude numbers that we have in these estimates. For the CDC's estimates, they took it out a very long time. They took it out to January. And what you get at that point is they're just getting unimpeded exponential growth.
Right.
And you have to ask yourself at some point, well, is that going to stop? I mean, you can't do that. We don't send our forecasts out that far. We only go 6 weeks out because we ask ourselves, well, isn't it going to stop? Aren't there going to be changes in the interventions? Couldn't there even be changes in the virus? Aren't there going to be more boots on the ground and a better control of it, which could radically alter how it's going to progress? And
won't there even be changes in the distribution of the population, maybe?
Absolutely. So, and that gets to the patchiness, the heterogeneity in space that exists within these outbreaks. You might have it burn out in certain areas, even possibly. And how that's all going to aggregate up to, let's say, a countrywide signal for Guinea or Liberia is really difficult to say. So, they put very broad error bounds on that. And the information is very useful in the sense of we need to take notice. And the more people who understand the alarming numbers that are potentially in the future as a result of this outbreak, and the more people who stop and take notice, maybe there'll be a better response. Certainly, we need to have the response earlier. The earlier, the better, because the more we do now, the more we can impede that growth and reverse the course of this.
So on your webpage, you have two graphs. You have infections versus mortality, and it goes out 6 weeks, as you said, right?
Yep.
And you can actually pick the country. You can pick an intervention, no change, degraded, or improved.
Right. Now, those are very loose characterizations. This is based on something a little nuanced that we observed. If we looked at the early outbreak stages prior to July, or maybe through the end of July, and say Guinea and Liberia, what we saw was a very erratic time series of incidents. And that could be due to problems with the reporting of the data. It can be due to differences in the spatial spread of it. But it also may reflect in part both the spatial spread and attempts at containing it being more or less successful through time. So in other words, it emerges in an area, it's recognized, and when it initially emerges, the growth is very unimpeded and aggressive.
Right.
And then as they jump on that area, it gets isolated and they have containment. Yeah. It depresses the growth in that area. And so that spikiness that we see early on is some indication that there's a lot of movement in the force of transmission, depending on how much efficacy is brought to bear in controlling it through whatever measures we can. So we did an early runs with it with our model, and that's hard to simulate with a very simple model. And we try to use simple models because you get complex, then you can't understand it, you can't constrain it. Bottom line is that we see some variability there, and that's what that expresses. We take the worst case scenario where it's degraded and the best case scenario where it's improved, and we use those as our force of transmission going forward. The no change just is the optimal projection that we have based on what's happened, but the other ones are assuming that there's either a breakdown in control efforts or an improvement. They're really speculative, they're guesses on our part, but they just give us a sense of, are we trending in the right direction? Because any deviation of our forecast from that, you could say that, well, the forecast isn't good, but you could also look at it and say, well, maybe in fact there's an improvement going on of control. So, in truth, what we're doing here, it's been very interesting for modeling, because in some instances modeling is not always paid attention to. But here, because there's so little information, people are desperate to know what's going to happen. We've never had this. We don't know what the end game of this is going to be at all. So anything that provides some sort of logical, sensible approach to looking into the future is really welcome. And we know there's a lot of uncertainty associated with it. Our credible intervals have to be very, very large. But if we can look at it and say, maybe if we do better this way, this will turn it around. We can get more sophisticated as we go along, but that's what we put on our website.
You also have a tab, which is data, which is the information, I guess, you collect from WHO, right?
The data is, so that's a table that you can hit instead of looking at it graphically. And what it shows you is the forecasts as we've made them each week in the past. And you can actually see how well the forecast did. So it shows the cases from the WHO. It shows our mean prediction, because we run an ensemble of prediction models. And then it shows the error associated with it. So in some places, we're not doing so well. In some of them, we are actually doing very well. Sierra Leone, the predictions have been remarkably good for the no-change scenario, which is, unfortunate, because it suggests that there's really no slowing of the exponential growth as we're estimating it.
So here you have the latest statistics, or September 21st, that you can compare to your predictions. And then after that, you've got predictions, but you don't have any numbers yet for WHO for the 28th. I guess they're not in yet.
No.
So you have to go manually and get these numbers and put them in here, I presume. Right?
We do. We're working off the cumulative incidence. There are also Ministry of Health reports we can go with also, but all these data are, we don't have a real sense of the error that's associated with them.
Now, you mentioned Sierra Leone seems to be kind of following your no-change prediction. The other countries, Guinea, Liberia, they are separating from that a little bit.
They're separating from it a little bit. So Liberia was on track with the no change for a while until a couple weeks ago. However, that said, this again gets to the issue of the data and how well it's reported. We've had a lot of discussions with other modelers and other forecasting groups and people at HHS and the CDC, and the quality of the data over the last 2 to 4 weeks is not really what it should be. When I say not just these last 2 to 4 weeks, but any last 2 to 4 weeks, it's not fully filled in. Our data, as we're using it off cumulative incidence, doesn't ever quite get that right, because we just pull it in. But the cases that are reported now this week may actually have been cases that really belonged in the previous weeks. Right. And if that were the case, that would push it up and push up the case count in, let's say, Liberia and Guinea, and make it more of a continuing exponential growth. In truth, we just don't know. We don't know. And, dealing with imperfect, noisy data, biased data, that's something that we have to deal with. It's just that, Ebola is obviously a very difficult, pressing need right now, where we wish we had something that was a little cleaner.
So things might be improving in Guinea and Liberia, or the data might just be worse there.
Those are two of the possibilities. Yeah.
So in past outbreaks, are there any hints as to how to conduct the current modeling based on the fact that in rural areas these things just sort of got contained, and we knew what the transmission rate was for those 3 or 4 weeks, or maybe even 5 weeks, but we know what the extended forecast was for those regions, right? In retrospect, we didn't know at the time, because it didn't spill over into the populated areas. Do any of those numbers mean any have any significance for the current outbreaks?
Well, they do. I mean, you have something that's a basis for comparison, and you can certainly have estimates of past basic reproductive number, how many infections an infectious person causes in a fully susceptible population. That's what the definition of that is. And that gives you an estimate of the force of transmission. And we can see that there are some similarities there. But it really is still uncharted territory because it's never gone on this long. It's never been this large. It's never been a bunch. I mean, essentially what you have is a bunch of populations that are interlinked with each other that are exploding with infections and then maybe having them clamped down.
Yeah, but I was going to go back to that small point about sexual transmission. At least you know that perhaps that's not a mechanism for transmitting based on the past epidemics.
We do. You know, we don't explicitly model that in our models. I mean, there's a lot of detail that you could put in, but because we don't know how people are interacting, we don't have anything, we don't have contact tracing patterns or anything like that. We're not going to get sophisticated and fancy with this.
But the assumption in the letter that we read was that everybody's going to have sex once they start to feel better, and this may not be the case.
You mean that they'll have sex, or that they'll be transmissive?
They're either feeling well enough to have sex and they don't transmit it, or they're not feeling well enough and therefore they don't have sex and therefore they don't transmit it. One of those two things has to be going on in the population where they've recovered from it, and there were hundreds of cases.
Well, I guess it's possible, but we just don't know. And there can be, you know, and the human response can be very heterogeneous also.
So it may be worth going back and looking into those populations to see what their cultural behaviors are.
Absolutely.
And then you can really learn a great deal about some behaviors safely, because those are zones that are no longer in transmission.
One of the things that's important, I mean, these are the types of things that are sort of, I don't want to use the word, but after the epidemic, I was going to say post-mortem, but I don't like the word in this case. Exactly. But after the outbreak, this is the type of investigation that ultimately has to be done. But right now, there are some really pressing data needs that really need to be looked into. And in particular, the mobility in the area is something that really needs to be understood. And it's an active area where people need to go. They need to get into the phone service records and find out how people are talking to each other and how they're moving about, because that provides information on mobility, at least within country, that we really need to see to get a sense of how things are spreading. That kind of data may be very informative in the here and now. And that's the type of stuff that we have to be judicious about looking for, because again, we need boots on the ground. We need nurses and doctors in beds, and that's the highest priority. But there are things that we could get right now that would really help that effort. I mean, where do you put them? How many do you know, they talk about making these smaller critical care units rather than the larger ones that they use right now, and maybe putting them in villages. But are they less secure? Is that the better optimal strategy for distributing it? We don't know. I mean, and that's the type of thing where if we had some better data and some better model estimates, we could look at those possible alternatives.
It really does help to know what the reservoir is in nature. Because the original outbreak that this is all based on is the Lassa fever outbreak in Nigeria, where the multi-mammate rat was eventually shown to be the animal of tolerance, sort of like bats and rabies. And only seasonally, when there were severe droughts following harvests, were there risks to people, because the rats were out of food outdoors and they had to go looking for it stored in the houses in the form of last year's grain. So you had these correlations that were pretty easy to match up once you understood the transmission. You're shooting in the dark right here. You still don't have a clue as to how this actually started in any of these outbreaks, right?
Well, yeah. I mean, I think some of the suspicion is that bats are the key reservoir for Ebola. I don't think we can say it definitively one way or another. We know that they do have it. Certainly what you have in West Africa, with the population growing and urbanization happening, and there is some level of deforestation, if you get fragmentation of the environment in which the bats or the rats or the primates live, you're going to have potentially an increase in contact. Now, the thing they worry about bats is that people fragment the forest, and then the bats all congregate in the mango trees that people have in their yards, and then you're going to come in contact with bat saliva and bat feces, particularly the saliva. They're very sloppy eaters, the fruit bats. And they leave their fruit on the ground and entrail and saliva, and they defecate on it. And so there's a lot of potential for contamination there. And of course, there's also, bushmeat trades that they're very concerned about. So the limited evidence that we have from phylogenetic analysis of this current outbreak suggests that there was one spillover event, but we don't know. And certainly the one that's going on in the Congo is thought to be a separate one, though.
Exactly.
I have a question, Jeff. I didn't look at the data table until just now during the show, and the numbers there in orange are training based on the actual data that you've gotten, and the blue is the forecast.
Yeah.
And so as we go down with time, there's more and more orange numbers. So
Right.
what I can't tell just from a quick look is, does the additional time of training make your model better at predicting in any of the segments or in the combined, or
Yeah, no, does it improve it?
Right.
Because, there are two ways of looking at it. One would be that maybe you need more data so that you can have better, more optimal constraint of your model and make a better forecast. We're not seeing a lot of shift in the more recent weeks, except that just sort of a slow change in the trajectory of the way things are going. So what we have is a situation where the additional data is just sort of nudging the model one way or another. And that's what we'd expect. There's continual adjustment going on, but it's curious because the predictions that we made for, let's say, Sierra Leone 6-7 weeks ago are very good, and predictions that we made then 8 weeks ago or 3 weeks ago are not quite as good. So there is some variation in what's going on, but what you have basically is really an adjustment of the exponential growth. Because it's in a fairly simple phase right now, unfortunately.
So some of these countries are recommending complete shutdown for a while, aren't they?
They tried it. Sierra Leone tried it last weekend. They had a 3-day lockdown, and it's very difficult to get a sense on how effective that was, but that's something that ultimately could be looked into, for instance, and see, retrospectively whether or not that had any impact on
But
it.
if they did, and assuming that they shut down the transmission, what an enormous effect this is going to have on their economy.
Exactly.
I mean, because you've got other effects that this plays upon, which are tragic when you think about how one thing snowballs into another. They're all interconnected in some way. I remember the yellow fever outbreak in New York City early on in our history.
You remember it? You were there?
I'm the senior member
You
of
have
this
no idea
group.
how old Dickson is.
And I can recall reading that a pastor said the only way to stop this epidemic is for us to all go to church. So they did. They all went to church, and they stayed there for about maybe 3 or 4 days. And the miracle happened. It broke the epidemic. But of course,
there were no mosquitoes.
There were no
in church.
mosquitoes in the church.
That's
Yeah.
exactly what happened.
Well, I think in this outbreak, whether or not flight restrictions or border crossing restrictions or the shutdown have an effect is something that really needs to be objectively studied.
Sure.
Certainly, people have talked about, the economic implications of doing this are far worse than anything Ebola could do. You can see things like that. I don't know the answer to that one way or another, but it's something that we'll be able to evaluate. There are other concerns, though. I mean, within these countries, there has been a complete collapse of the medical infrastructure, and it may have been limited to begin with, but at this point, the hospitals are either shut down or acting as Ebola wards, or in a very limited capacity handling births. That's it. That's all that's going on, at least for Liberia, I believe, is the one that I saw the statistics on. And, they're going into malaria season there. And malaria is their biggest problem in that part of the world. And so what is that going to mean when they have their usual surge of cases and problems associated with that, particularly among children, if they don't have any infrastructure?
Here.
So you said really the only data we have are the cases, the deaths, and so forth. But the CDC has used other aspects of that to try and model. They've used the available beds, how long infectiousness is, and so forth. Do you use that also in your?
No, we don't, because I'm a big proponent of model simplicity, parsimony, and putting that in there and projecting it into the future is putting a scenario in play. And I don't know how that's going to unfold. We certainly can do it, and we've thought about doing it, but we have a lot of reservations about putting that into play. And I'm not poo-pooing it and saying that it shouldn't be done, and CDC has done it, and other organizations have done that in theirs. It just adds another level of uncertainty into what you're doing, into the projections that you're making. And we just decided to keep it simple and just focus on what's going on with the cases.
So the CDC at the end concludes not basically they say none of this could be right, because it's full of assumptions. How should we look at 500,000 to 1 million?
Yeah. Well, and you say to yourself, are they washing their hands of it or something along those lines? What it is it's an extension of the exponential growth that we have right now. We certainly have plenty of people in the area to support that kind of level of an outbreak. Even the 1.4 million they said is their upper bound. There are enough people in the region that is possible.
Yeah.
What's going to dictate whether that happens is how we as an international community respond to this and whether we really clamp down on it. We do know what to do to deal with Ebola. I mean, that's something that we've always known, and that unfortunately that may have been a hubris that fed into this outbreak, where we thought, well, we're not going to have a problem with it, we'll figure it out. But I've talked to people at Doctors Without Borders, and they say it was in May or March, one of the M months, that they knew that this was different. They knew that they had something on their hands that was not quite the same as what they dealt with before, simply because of where it evolved.
Exactly.
So what needs to be done to curtail it then?
It needs resources. It needs people on the ground there. It needs the nurses, the beds, the doctors. It needs the security. It needs the ability of people to go out and do the contact tracing. It needs all that. It's grunt work is what it needs. It needs the grunt work on the ground. And it needs it on a scale that's never been implemented. And that's the thing that's a little frightening, because the question is, can we actually do that as a community, an international community, put that together?
So what it needs is a functioning medical infrastructure and government.
Yeah. And it needs to be imposed.
The absence of which is pretty much what caused this.
Yeah, you could argue that. You could certainly argue that.
Is it possible to impose that on the countries?
I'm well out of my area of expertise here, but
yeah, so are we.
But I mean, I don't know what the alternative is. I really don't know what the alternative is.
You've got.
I'm pretty sure what you would be told if you invited any of the people in positions of power onto the show and asked them what they feel about nation building in West Africa.
But it's not the only area where you've got problems, though, Alan, and you know this too, because we've already discussed, like the polio workers in Syria, for instance, that were attacked and killed, basically. So there's a tremendous mistrust on the part of some segments of the population to interveners. All these do-gooders, they're actually not do-gooders at all. They're trying to take advantage of us.
Well, in the countries that we're talking about here, Guinea, Liberia, especially Sierra Leone, there's a mistrust that is entirely well-founded.
Oh, no question.
I mean, these are places that for decades, if somebody said they were the authorities, you ran like hell.
Yeah. That's right.
So, Jeff, I'll ask you one more question, then we'll let you go.
Mm-hmm.
How is Ebola infection transmitted?
It's transmitted through bodily fluids, as we understand it. It's contact. We don't have a lot of evidence that it's aerosolized at all and that it's airborne. I haven't seen anything like that, and I don't think that is the way of transmission. It's not going to be transmitted by mosquitoes any more than HIV is. You need to be highly evolved to the mosquito's life cycle if you're going to be transmitted by a mosquito. It's by vomit and diarrhea and blood and sweat. That's what we see in the literature. That's what we understand. It's that type of personal protective equipment that they put on when they work in these field units, and they don't get sick if it's used properly.
Okay. Anything else for Jeff before we let him go?
All right. This is great.
This is excellent.
I really appreciate it.
Yep, my pleasure.
I think we'll probably have you back before this is over.
All right.
And at some point, we'd like to talk about what other things you do in your lab as well.
All right.
Climate modeling. I want to hear more about that.
All right. Well, we can talk about the weather ad nauseum. All right. Thanks for having me
Thank
here.
you, Jeff. Really appreciate it.
Thanks so much.
Okay, Dickson, are you still there?
You bet. Absolutely.
All right, I want to go back to...
I'm proud to say that I was a member of that department for some 20 years.
Yeah, you were.
So that's
no, he's...
interesting.
We actually had a little interaction a few years ago. He's got an interesting sampling study going on, which hopefully he'll talk about one day. Back to the follow-up. Hey, yeah, we actually have a few other things to talk about. Letter from Scott, who writes... Vincent, the other day I listened to your interview on NPR in which you seemed quite confident that the risk of spread of the Ebola virus by aerosols is currently non-existent and that status is extremely unlikely to change via mutation. I would like to direct your attention to the following article which suggests that indeed there is evidence, however weak and as yet unconfirmed, that Ebola may be weakly transmissible via aerosols. And he links to a article at CIDRAP, the Center for Infectious Disease Policy and Research, by Brosseau and Jones, who is called Commentary, Health Workers Need Optimal Respiratory Protection for Ebola. And Scott continues, perhaps you should review this and correct yourself, at least via TWiV, if not via NPR. Scott is in Costa Rica. Now, as you heard Jeff just say, Jeff is an epidemiologist. There is no evidence that there is an aerosol transmission component. This article suggests that there might be, and it is speculative. And I note that it's not in a peer-reviewed journal. And I would also like to note that the head of CIDRAP is Michael Osterholm. So I think that there are interesting discussions in this article, but the main mode of transmission is clearly not via aerosols. So I would submit that no correction is necessary, and that what I said on NPR stands.
We still don't have any evidence of this virus being transmitted through the air.
Correct.
No, nor does CDC, and they list this as the only legitimate way it can be transmitted. So
Right.
if this listener is taking exception to Vince's testimony, he's merely basing it on the official stated mode of transmission from the Centers for Disease Control and Prevention.
Well, I've also read quite a few papers in the
Sure.
last few weeks, and every one of them starts out by
Sure, exactly.
there's no evidence and we can't find it. In fact,
As soon as there is
come on.
evidence, though, we'll be glad to present it.
I mean,
Absolutely.
I'm a scientist. I tell
Hear, hear.
what's out
That's
there
right,
in the
that's right,
literature,
that's right,
and
that's
I would never say there's no evidence if there were.
And the basis of this commentary at CIDRAP is basically, well, it could, it might, it's possible.
Right.
And sure, anything is possible.
Yes.
That's not really a fair way to frame a scientific question.
That's right.
But it doesn't. In the experience that we've had so far, and in the experience that has been documented in this outbreak, even though this outbreak is unusual in many ways, nobody has yet documented a clear case of aerosol transmission. I mean, the people who are getting it
Sure.
are getting it because they didn't have the mask on and the patient vomited on them, or they were a family
Yeah,
member
that's
caring for someone who was dying.
That's right.
It's very clear contact transmission that's going on.
In this article, there's just one thing I'd like to read where they say, quote, "The important points are that virus-laden bodily fluids may be aerosolized and inhaled while a person is in proximity to an infectious person, and that a wide range of particle sizes can be inhaled and deposited throughout the respiratory tract," end quote. There's no citation for this. You know, the point that this could happen, again, is just saying this may happen. There's no data.
Yeah, as we'll see if we're going to talk a little bit about this. If you make an aerosol with Ebola virus, you can infect animals with that aerosol, but typically those aerosols are made by breathing, talking, coughing, and for aerosol transmission, the aerosols travel for a distance, quite a distance, so that's not what's happening here. All right, Alan, can you take Kevin's?
Kevin writes, "Greetings, Doctors TWiV. On the most recent TWiV 303, Born of This Way, while discussing the quirks of bats in relationship to viral infections, Vincent brought up the duplications of genes involved in oxidative stress due to the higher metabolism, which got me thinking about a paper my graduate lab recently published about the relationship between mitochondria, peroxisomes, and antiviral signaling," and provides a link. The lab had previously shown that cytosolic RNA sensors, RIG-I-like receptors, can engage their signaling pathway from two locations, mitochondria or peroxisomes. The take-home from this new paper is that signaling pathways engaged are different. Signaling from mitochondria induces type 1 interferons, while signaling from peroxisomes induces production of type 3 interferons. Not much is known about the difference between type 1 and type 3 interferons, since until now, everyone had observed them as co-regulated, and the dominant type 3 isn't expressed in mice. But one interesting thing this paper showed is that as cells get more peroxisomes relative to mitochondria, they increase type 3 interferon production, and if you eliminate mitochondria from cells entirely, you get a dramatic increase. Back to the bats. Their altered metabolism almost certainly affects their mitochondria to peroxisome ratio, and this could lead to dramatic differences in their antiviral responses. This could also explain why they're more tolerant to a lot of viral infections that we're not. Does anyone know if this is more often the case with RNA than with DNA viruses? And Kevin says, P.S. Audio Immunity, tagline, a podcast about our body's never-ending fight with the outside world, recently hit double digits. We're almost to Vincent's magic number. He once said that when you hit episode 12 with podcasts, it suggests that you're here to stay, since people who aren't serious will quit well before then.
Hey, good going.
Congratulations. Yeah.
Congratulations.
Very nice.
So many podcasts that I've talked about have gone down the tubes.
Yep.
Audio
Well, they
immunity.
stopped.
I like it.
I mean, my student Ashley's podcast stopped at about 8.
Yep.
There was a cool one about agricultural microbiology that stopped. So, I mean, it's great. Keep going. I remember talking to Lin-Fa Wang about this. He says he finds both DNA and RNA viruses in bats. And I remember he said tons of herpes viruses. So it doesn't, it's not a quantitative statement, Kevin, but they seem to tolerate DNA viruses as well. It's an interesting idea, however.
Right. This idea that the peroxisome mitochondria ratio might affect their immune response. That's cool.
All right, our last follow-up. Kathy.
Steve writes, "Hi, Vincent and team. I don't know about your American varieties and fridge temperatures, but here in England, if one tries to grow tomatoes outdoors, there's always a bit of a gamble how long to leave them outside, because the first cold night has them all on the ground. And if you bring them indoors, even though they may initially look fine, they rapidly rot instead of ripening. If, on the other hand, you bring the whole vine indoors and hang it up before the cold gets to the fruit, it will be fine for months. I've sometimes kept tomatoes in a bowl from one season until the next. The last small ones of the year that ripen slowly in the cool, like this, always taste much better than the ones picked in the height of summer. A similar situation is the transport of soft fruit like peaches. I used to deliver organic fruit and veg and had to pick up from the wholesalers. It became clear to me that the fragile fruit like peaches and nectarines were pulled off the trees none too carefully and then rapidly chilled before the bruises could develop. Right. Thus we would deliver fruit that looked great but was in fact dead and bruised. As soon as it warmed up, it rotted, and unless you ate it right away, you ended up throwing it away. Right. This meant that if the fruit had been picked not quite ripe and customers unknowingly waited for it to ripen, they just had to watch their expensive organic produce go to waste. I used to get quite outraged by this dreadful waste when organic and sustainable were supposed to go together, yet this stuff had traveled around the world just to be thrown away, thanks to negligence or just plain don't care. I've not read the chef's piece that you referred to, but it would surprise me if he was able to keep any tomato once it had been in the fridge. I'm quite sure that my own fridge would kill most of the tomatoes I've seen. I wonder what he has in his fridge. Okay.
Maybe.
That potato salad has gone very bad.
South, as it were.
Yeah. Incidentally, while on this subject, don't buy shiny tomatoes or peppers. They are not ripe and are still swelling. When they are ripe, they become duller and maybe a little wrinkly. For peppers especially, they tend to get sold off cheap when they are wrinkly and dull. They may even be thrown away. But this is when they actually taste the best. I would never buy shiny fruit, and I've picked up many a tasty bargain as a result. And I never put fruit in the fridge unless I'm actually freezing it. All the best, Steve.
Wow.
Everything you buy in the supermarket here is shiny, isn't it?
Yeah. That's to attract you to it.
Yeah.
Yep.
Bartlett pears.
But you know, my peppers in my garden are pretty shiny. I guess he would say they're still growing, right?
They
They're
still
not ripe
are.
yet.
Right.
The one that I've noted most is the green Bartlett pears that you buy when they're not ripe, and then you take them home and you expect them to look perfect because the green version of it did. And as they yellow, all the bruises start to come out, and it's really easy to see the difference between a carefully boxed bunch of fruit and ones that are just thrown onto a pile.
All right. Back to Ebola.
Right, exactly.
There's an excellent article in New England Journal this week, a summary of the outbreak, which we'll link to. It's really well written, and I highly recommend it. The author is Jeremy Farrar and Peter Pio, Pio. The Ebola Emergency Immediate Action Ongoing Strategy. And I just wanted to quote one line from it, which is, despite the vast scale of the current outbreak, the clinical manifestations of Ebola virus disease, the duration of illness, the case fatality rate, and the degree of transmissibility are similar to those in earlier epidemics. It is therefore unlikely that the particularly devastating course of this epidemic can be attributed to biologic characteristics of the virus. It is more likely to be a result of the combination of dysfunctional health systems, international indifference, high population mobility, local customs, densely populated capitals, and lack of trust in authorities after years of armed conflict.
Yep.
It's perfect, right?
It is.
That is very, very well put.
They say the outbreak was an avoidable crisis.
Yep.
How about that?
This is not a technology problem.
No.
All right. Now, in just 10 minutes or so, I thought we should revisit this issue of aerosol transmission. I went back in the literature and I pulled all the papers I could find where they talk about transmitting Ebola virus to animals. And, of course, you can't really study this in people. You have to make epidemiological observations if you want to see how something is transmitted. And as you heard, there's no evidence for it. But this is confusing a lot of people because, as we heard earlier, people are saying, well, isn't Reston aerosol transmitted? Why isn't that important? So here's what we know. 1995, a paper entitled Transmission of Ebola virus Zaire strain to uninfected control monkeys in a biocontainment laboratory. All right, this, where was it published? I just closed the window. Lancet. In this experiment, they infected rhesus monkeys intramuscularly with the Ebola virus. They died 7 to 13 days after infection. A separate set of control monkeys in cages that were 3 meters away became infected 10 days after the other monkeys died. Okay, so they were controls for an experiment that was ongoing. They got infected. The authors conclude the most likely route of infection was aerosol, oral, or conjunctival exposure to virus-laden droplets secreted or excreted from the experimentally inoculated monkeys. So this is basically a lab accident, or I should say controlled accident. The monkeys were not supposed to get infected. It wasn't a controlled transmission experiment, but somehow these monkeys got infected. That could be that the other monkeys threw stuff at them. Who knows, right? So, but that's the only incident where I can see that that there's suggestion that this virus went by aerosol from one monkey to another. Another paper, 1995, lethal experimental infections of rhesus monkeys by aerosolized, is that the name of it? Ebola virus. Well, that's my title. The actual title of the paper is Lethal Experimental Infection of Rhesus Monkeys with Zaire Virus by the Oral and Conjunctival Route of Exposure. They infected macaques with an aerosol of Zaire Ebola virus in a head-only chamber. So they put a chamber around the head. They made an aerosol of the virus, put it in. Guess what? The virus can infect the monkeys. It causes fatal disease. And we know that the mucosal membranes can be infected. There's a lot of literature out there saying this virus can infect the respiratory mucosa. Of course, if you put an aerosol right on the head.
In your eye, it could even go through the eye.
Of course. They found viral antigens in the airway epithelium. They found them in immune cells there in the epithelium. They could see particles way down in the lung. They did not see if the virus would transmit by aerosol from monkey to monkey. And they speculate in this paper why we don't see aerosol transmission among humans. In the discussion, they say maybe there's not enough virus. Maybe the temperature, the humidity conditions in the field aren't adequate. They say, quote, copious extracellular Ebola virus antigen was present on the mucosal surfaces of the nose, oral cavity, and pulmonary airways of aerosol-exposed monkeys, and strong evidence to support the potential for secondary spread by aerosol. So there's a lot of virus in the monkey lung when you infect them this way. Okay.
But this thing with the head-only chamber, I mean,
yeah,
this is one step shy of injecting it.
Right.
Exactly.
So you're putting a tight chamber over the head of the monkey, and you're spraying this aerosol in the monkey's face in this enclosed space.
Right.
It's a loaded deck of cards.
It is extremely stacked.
Yeah.
The next paper is 2011. Aerosol exposure to Zaire Ebola virus in 3 non-human primate species, differences in disease course and clinical pathology. This was basically a study in microbes and infection to figure out what animal would be the best for these kinds of infections. So, they do an aerosol exposure just like the other one in a head-only chamber. They do cynomolgus, rhesus macaques, and African green monkeys. They're all infected. But they didn't check to see if the virus could be transmitted by aerosol from one animal to another. A 2011 paper in the Journal of Infectious Diseases, replication pathogenicity, shedding, and transmission of Zaire Ebola virus in pigs. So Reston Ebola was found in pigs in the Philippines. So they wanted to say, well, what's with pigs and other viruses? So they inoculate these animals with with Zaire Ebola virus by dripping virus in the nose, in the eye, and in the mouth. Okay, all at the same time. So you don't know which route is working, but every pig, nose, eye, mouth. Virus reaches very high titers, 10 to the 7th infectious units per milliliter in the respiratory tracts. Wow. Pigs develop severe lung pathology, and they can transmit the virus to co-housed pigs. All right, and they say this is not a proper aerosol transmission experiment because we can't tell if it was aerosol or contact because they're in the same cage, right? Sure, so they could be contacting each other. So again, the virus can infect the respiratory mucosa. These pigs, however, they note, develop severe respiratory disease that we don't see in people infected with Ebola virus. Okay, now perhaps if you inhaled as much as they dripped in the nose, you might see it. But in these animals, they're making a lot of virus, and there's a lot of pathology in the lung. Another paper published in 2013, Pathology of Experimental Aerosol Zaire Ebola Infection in Rhesus Macaques, Veterinary Pathology. They infected the monkeys with, again, Ebola Zaire in a head-only chamber. The virus replicates in the lymphoid tissues of the respiratory tract, spreads to regional lymph nodes, it replicates, then you get a viremia, and that brings it to other tissues. The virus titers within the lung are modest, 10 to the 5, 10 to the 6 infectious units per milliliter. They don't actually see virus in the lung epithelium. They don't look for the virus in respiratory secretions. They don't test for aerosol transmission. Another paper from 2012, they tried to use mice. They tried to do aerosolized Ebola virus in mice. Basically, they put the whole mouse in an aerosol chamber and introduced the virus aerosol. And again, these are aerosols generated by a machine, right?
Right.
So instead of a head-only chamber, this is a whole mouse chamber.
Mouse chamber.
Yeah.
And again, they generate these aerosols artificially in a machine that's making little droplets with virus in them. Here, the virus does not replicate in wild-type mice. They have to use immunocompromised animals in order to get it. So the mouse is not a great model for looking at this.
Or they have to use mouse-adapted Ebola virus.
Right. The next paper has been mentioned quite a bit, and I just wanted to give a few more details. This is a paper published in 2012, Transmission of Ebola Virus from Pigs to Non-Human Primates. It's in Science Reports from Nature. And here they infect pigs oronasally, like we just talked about. They drip the virus in the nose. And this, of course, is done in a BSL-4 lab. So then they put the pigs on the floor in this laboratory, and they have a picture of these pigs. The floor, those little it's a rubber floor with those little circles to help improve traction. Right? The pigs are just kind of walking around on that floor. They're infected. And then next to them, there's a stack of cages where they put macaques. Okay? So the pigs, of course, get infected. And then the macaques get infected. All of them got infected, and they can find virus in the macaques, although not much pathology in the lungs. And they write, transmission between macaques in similar housing conditions was never observed. We know pigs shed a lot of virus and respiratory secretions. And they also write separately, the design and size of the animal cubicle did not allow us to distinguish whether the transmission was by aerosol, small or large droplets in the air, or droplets created during floor cleaning, which landed inside the non-human primate cages, fomites. So they note that every night someone had to go in and clean the floor because you can imagine.
There are pigs walking around on it.
Yes. Pigs are going to urinate and defecate, right?
Yeah.
Wow.
I mean, so this is the thing. Everyone cites this as transmission by aerosol, but I think it's kind of not proven, right?
You put the monkeys in a pigsty.
Right.
And they got the virus.
And one other thing it proves is that the BSL-4 facility actually works.
Yes.
Because the workers didn't get infected.
Yeah, I'm choosing them to go in and clean. They had to wear the BSL-4 suits, right? Of course.
Sure.
So the pigs can aerosolize perhaps the virus, and they can transmit it by some way to macaques. But the point here that I want to emphasize is that macaques don't transmit it. If they infect the macaques separately, they don't transmit by aerosol to one another.
Right.
And I've been trying to emphasize that in many of the studies, there's not a lot of virus in the lung of the non-human primates, and that might be related to it. A 2014 study entitled Evaluation of Transmission Risks Associated with In Vivo Replication of Several High Containment Pathogens in a BSL-4. Here they took a bunch of different dangerous viruses and infected a variety of animals with them. They infected in the one that we're interested in, rhesus macaques intramuscularly with Ebola virus, again, Zaire. They put uninfected animals in cages nearby, 3 feet away. The infected macaques, again, they're IM inoculated, intramuscular. They had virus in their nose and nasal swabs, 4 to 5 logs of genomes per mil. The nearby macaques did not get infected. So they conclude airborne transmission of Ebola virus between non-human primates does not occur readily. And they say what they should do is try to infect the donor macaques mucosally, right? And that would maybe generate more virus in the mucosal system. And then the last paper is actually a study of an outbreak in 1999 in the Democratic Republic of the Congo. 1995. The paper was published in 1999. The title is Ebola Hemorrhagic Fever, Kikwit, 1995, Risk Factors for Patients Without a Reported Exposure. So here they had an outbreak of 316 people, and they could go and do tracking like Jeff was talking about. They could go to ask these people, who did you contact, what did you do, did you touch, whatever. And they said for 12 of these patients, they couldn't figure out what the exposure source was. All the others they could say, they were in contact with someone. So they say, well, maybe it was airborne. Actually, what they say, it could have been touch, droplet, airborne, particle, or fomite. So a lot of people do cite this paper as saying evidence for aerosol transmission, but in fact,
this is really an absence of evidence.
It's absence of evidence.
Exactly.
It's absence
of anything.
Evidence. So I tried to. Someone asked me, "Is there virus in the respiratory tract of people?" And of course, you can't. Whenever there's an outbreak, it's very difficult to study because they're in remote areas. The current outbreak, of course, nobody's studying the infected people because they're scrambling just to save everyone. The virus, as we said, has been found in various secretions. It's in saliva, okay? It's in vomit, and semen, and feces, and etc. But is it in respiratory secretions? No evidence for finding it there. And that's what you would need for getting high efficiency aerosol transmission. So, I mean, my conclusion from all this, you can give Ebola Zaire to animals by aerosolizing the virus in a machine and giving it to them very close, but it doesn't transmit by aerosol among them. I would bet it doesn't get the high enough titers in the lung, and as at least one reason, but there are probably other reasons as well. So, and again, I don't think it would change because we've never seen that happen before. It's highly unlikely. So,
and this shows that the lack of evidence for aerosol transmission isn't because nobody's looked.
Yeah, people have tried.
Because these are heroic efforts to
yeah.
try and get some kind of aerosol transmission of this virus, and it doesn't work that way.
Just a couple of quotes from ProMed Mail. I was looking through. There's a letter from Professor Mitar at South Africa. I am rather concerned that we have lost perspective of the mode of transmission in this Ebola outbreak. To my knowledge, it is not aerosol-borne, so I am at a loss to understand why there is so much emphasis on this route of transmission, when clearly the need is to reduce splash and droplets in the 40-50% of cases where respiratory bleeding is found. And then the moderators at ProMed Mail agreed with that. And lastly, last time Alan mentioned the article I wrote about the chance that Ebola virus might transmit by aerosol by mutation in this epidemic. I had said not likely. And I just want to note that Ben Newman, a Reading virologist, says there's no solid evidence that aerosol transmission occurs. Ian Jones, a virologist with the University of Reading, said, we've never seen a human virus change the way it's transmitted. They actually cite my article, which is quite nice. And then Tony Fauci at the NIH, very, very rarely does a virus completely change the way it's transmitted. I don't know what even very, very rarely is. But
Yeah.
Tony has to be careful, right?
He's allowing the possibility that somebody might pull one out.
So that's the story there. What do you think? Should we go right to picks?
I think we're at an hour and a half.
Yeah,
right?
let's go to picks.
Yeah, we should do an all email episode sometime.
Yeah, because the next email is quite long. Let's go to picks of the week. I think this was a comprehensive episode, right? We covered a lot of ground.
And we covered a lot of email that was follow-up
Yeah.
email.
Yes.
So
All true.
And it's just more and more of that at the beginning.
Yep.
Kathy, what do you have for us today?
I have a pick which follows our arc of microscopes, and there's competing evidence now from Alan and Rich as to whether this has been picked before, but this particular link was from Phil, someone here at U of M who sent it to me, and it quotes a blog from head of NIH, Francis Collins, of a print and fold origami microscope, and he had seen it demonstrated at some White House science fair-like thing, evidently, and was quite impressed with it. And so there's also I provided a link to the PLOS ONE article that's open access. Everybody can read it. It's very technical, I have found, in a quick skimming of the article. But you basically print this thing out on cardstock, fold it up, use some kind of glass or something or other to be the optics of it, and you can look at things. And so it's being promoted as things to be used in developing countries. There's pictures of children looking at it as if at a science fair, and
They're looking at parasites, Dickson. Look.
yeah.
I can't see it from here.
Plasmodium, trypanosome,
Look at you.
Giardia, and Leishmania.
My goodness.
Cool.
That's got high resolution then.
Do you
Yeah.
think?
I do
Yeah.
think.
And this is actually very reminiscent of Leeuwenhoek's original design.
How about that?
Yeah, look at that.
It's the card, and you put a little glass bead on it, and that's your lens, and that's pretty much how Leeuwenhoek built his. And you hold it up to a bright light source and look at your sample, and it takes a standard microscope slide.
This is great. So this guy visited a mosquito-infected rainforest in Thailand and found a clinic with a $100,000 microscope that was unused in a locked room.
Yep.
So he said, "We got to do better."
Yep.
This is great.
Yeah.
50
So
cents.
in Francis Collins' blog post, there's links to a number of things, including to a TED Talk. So you can look at this in whatever media format you like to get this kind of information.
That's cool.
Yep.
Ellen, what do you have?
I have an old book. I would have provided an Amazon link, but this is long out of print. And I was just looking through it again recently, looking through my copy, because my daughter's gotten an interest in collecting insects. No idea how she picked that up. But so this is Henri Fabre's Book of Insects.
Huh.
And the specific one that I have is actually a translation from the original's in French. This is a translation that was published in the 1930s. And it has illustrations by an artist who drew a lot of the scenes that Fabre is describing. If you're not familiar with Henri Fabre, he was a naturalist, French naturalist who, 19th century, did some really pioneering studies on insects and looking at life cycles and figuring out why they do particular things. And he really just started out at that time. You didn't become a scientist without being born into a wealthy family, and he was working class. But he, as a kid, he played in the fields and he observed the bugs, and he managed to actually parlay his knowledge of insects into a very prestigious academic career. So his story is cool, and this book is cool, and one of the really neat things about it is the artwork.
These are fabulous pictures. If you click on some of the eBay entries, the second one, or the one that's $39 and has 6 pictures, there's a couple of really good ones there.
Oh, look at that. Nice. Beautiful.
Yeah.
Well, there's one for $8, huh?
Yeah, there's a range
so
of prices.
I mean, I'm just giving a link for eBay, and obviously usual caveats for eBay apply, but You can probably shop through
cool.
and find good condition.
Dickson, what do you have?
Well, I'm sure I haven't picked this before, or at least I don't think I have, but according to my memory, that's no judge. I picked the Landsat website for NASA for Landsat 8, which has only been in orbit for about a year now. I'm not sure if I picked it before, because if I did, then I take it back and I'll have to pick something else like Lady Rothschild's photographs and pictures of fleas. But to follow up with Alan, because actually she does have a collection of them, and it is quite remarkable. But the Landsat 8 satellite has improved the resolution of Landsat photographs of the Earth to the point now where we can tell the stage of development of plants as to whether the crop is ready for harvest or not, or whether there's been a blight of something like wheat rust or rice blast, or whether there's going to be a good or bad year for, you name the crop, and it can do that, as well as document, of course, the loss of hardwood forest due to deforestation. So, I mean, we've got a big environmental movement on foot right now, that just began about 3 weeks ago and culminated in some protest marches over the last 3 or 4 days as the result of the United Nations meeting here. And even they are now getting on board for climate change issues and learning how to talk about it in a way that makes sense. And here's the data. You might not believe what you hear, but you can certainly believe what you see. And so Landsat 8 lets you see exactly what is going on. There's no way to hide from Landsat 8. That's the point. So that's probably why I picked it.
Yeah, you didn't pick it before. I just checked.
Thank goodness.
It's good.
It has an image of the King Fire in California.
Yeah, and you can almost read a newspaper from outer space with this one. It's that kind of a resolution instrument, and it's quite remarkable.
Nice.
My pick is a very short post called "Some Advice from Jeff Bezos," or Bezos. I don't know how you say it.
Bezos, I think it is.
And it's based on a conversation he had with this blog poster, and he said, people who were right a lot of the time were people who often change their minds. He doesn't think consistency of thought is a particularly positive trait. It's perfectly healthy to have an idea tomorrow that contradicts your idea today. He has observed that the smartest people are constantly revising their understanding, reconsidering a problem they thought they'd already solved. They are open to new points of view, new information, new ideas, contradictions, and challenges. This doesn't mean you shouldn't have a well-formed point of view. It means you should consider your point of view as temporary. I guess we should add to that a well-informed point of view, right, Alan?
Yes.
Anyway, this is a good way to be a scientist, right?
Indeed.
Mm-hmm.
That is the
flexible.
That's the definition of a scientist, in fact.
So I thought that was cool.
Great.
We have a listener pick from Justin. He sends a link to an article. Brazil releases good mosquitoes. This is an article in BBC News, and the BBC person watched as they were released. There's a video of that here. Brazilian researchers in Rio have released thousands of mosquitoes infected with bacteria that suppress dengue fever. I wonder what those bacteria are, Dickson.
Duh. What are they?
You don't know?
Tell me.
Wolbachia.
Wolbachia.
We've done TWiVs on this before.
Okay, then.
Yeah, it doesn't. For dengue, it actually inhibits viral replication.
All right. And Plasmodium, too, by the way.
It does other things to the mosquito,
right,
in terms of its reproduction and so forth. And I think we talked about this paper on TWiV a while ago, that it does have a direct effect on dengue replication. And so there was a release in Australia previously that we talked about. So now this is in Brazil, which is where I'm going on Sunday.
Right. Look for those mosquitoes.
Well, this is Rio. I'm not going to be in Rio. How far do mosquitoes fly, Dickson?
Depends on the species.
Okay.
Some of
Depends on whether they get on an airliner.
Exactly right.
Can they do that?
The salt marsh mosquito, of course they can, but the salt marsh mosquito of New Jersey can fly over 50 miles. That's a flight range. That's a pretty big flight range.
Wow.
Yeah.
Dickson, did a mosquito fly from Israel to New York City?
Yeah, but via airline. No. Actually, the virus did that.
In a person?
Yeah, probably. 99% sure.
All right.
Mosquitoes can also travel on ships, of course.
They can, and cars,
And
and trains.
Yeah,
and the eggs can travel on tires
without paying, right?
Yeah, wasn't the Flying Dutchman story about, yellow fever and mosquitoes on the ship.
There you go.
Something
There you
like
go.
that.
So we introduced the Asian tiger mosquito this way by taking old truck tires, shipping them
Right.
to Japan, having them retreaded, and then brought back to the States. And while they were in Japan, they accumulated water from a rainstorm, and the Aedes albopictus laid its eggs along the edge of the water level, and then they shipped those tires back to the States, and some of them hatched, and the next thing you know, we've got the Asian tiger mosquito here.
The next thing you know,
The next thing you know.
that's TWiV 304, Dickson.
Great.
And the sponsor of this episode has been the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. It's a great virology program. Go check out their PhD program if you want to get one. They have a deadline of December 1st. Go to mssm.edu/MIC and see if it's for you. Maybe you apply and you work with Peter Palazzi, you do a postdoc with David Baltimore, get a job at Columbia when I leave, right?
Could be
Dickson?
the next
There you go.
podcast hoster.
I was going to say, and then you do a podcast.
And then maybe you train a PhD student, they become a science writer.
That's right. Oh, they could very well be that.
Well, Dickson, that is Alan, you know.
Yes.
I don't know if you understand when I make these oblique things.
No, that wasn't very oblique. Thank you.
Okay. This episode and all the others can be found at iTunes and also at TWiV.tv. And we love to get your questions and comments. Send them to TWiV at TWiV.tv. Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
Pleasure.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun, and it was really nice to have Jeff on too.
Yes,
I enjoyed that.
Indeed.
Jeff Shaman is a professor here at the School of Public Health at Columbia University Medical Center. So, Dickson, that's a separate school, right?
It is. Yes, it is.
But it's part of Columbia,
correct.
and it's at the medical center.
Yes.
Right. So they have their own dean.
They do.
They have
Their own budget.
their own money.
They have their
That's
own
right.
mailman.
They have their own
It's the
mailman.
Mailman School of Public Health. Who is Mailman? A person that gave money,
Yeah.
Dickson?
Yeah,
Yeah.
he's a very wealthy guy.
So their dean doesn't have to answer to the medical school dean. Is that correct?
Well, they're all part of a consortium of the Health Sciences Center. There is a super
Did you
dean.
say conspiracy?
No, consortium. Consortium, please.
I just misheard you. I'm sorry.
Yeah. No, they have meetings, and I think, in truth, the medical school dean still rules the roost in terms of what actually happens here.
Kathy, is there a public health school in Michigan?
There is.
Oh, a
It's
very good
in
one.
fact where some of that poliovirus work.
The Rackham?
Yeah. Well, no, that's the
where
graduate
they-
school's name and the building.
That's just where they presented the data.
But Tommy Francis and
Yeah, they were in
so
that
forth
school?
and so on. No, they were in the public school of public health.
So that's a separate school at
It is.
Michigan, right? Okay.
Yes.
And they have their own dean as well?
Yes.
Oh. Interesting. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.